So let's see what are the physiological buffers, how effective they are, and what is their pKa. So for understanding how effective physiological buffers are, we should know that what will happen if pH is less or more than the pKa of the buffer. Obviously, because it is governed by this equation, if pH changes, the concentration of these dissociated ions and the undissociated acid will be different, right? Only if pK is equal to pH, the concentration will be equal. However, if pH is changing, the dissociation of the buffer will change and the concentration of the conjugate base and the undissociated acids will be different. So that is what is happening with physiological buffers. See, normal plasma pH we said is 7.35 to 7.45 and buffer is most effective in either direction when pK is equal to pH. But that is not the case with physiological buffers. Our physiological buffers, that is like H2CO3, have a pKa of 6.1, while another buffer which acts intracellularly, that is the phosphate buffer, its pKa is 6.86. On the other hand, ammonium buffer, which is present in tubular fluid in kidneys, its pKa is 9.25. So, because the pKa of the physiological buffers is much different from that of pH, they are not effective equally in either direction. And that is good actually, because we want that physiological buffers should be more effective for acids, since it is the acids which are added a lot due to various activities, cellular metabolism in the body fluids. Yeah, bases are also added, but compared to acid, their concentration is much less. So, suppose H2CO3 dissociates into its ions, that is hydrogen ions and bicarbonate ions, and the pK is 6.1. So, that means the concentration of these dissociated ions will be much more. So, whenever the pK is less than the pH, we get the dissociated ions more. So, obviously they will combine with the hydrogen ions which are released by the acids, and this H2CO3 will form. Now, this buffer has one added advantage, that this bicarbonic acid dissociates into carbon dioxide and water, and it is this carbon dioxide which is exhaled out by respiratory system. So, this helps us. Why? Because even if lot of acids are added and a lot of H2CO3 will form, it will prevent the accumulation of H2CO3. You might have read in chemistry that if the products accumulate a lot and if it is a reversible reaction, the reaction will start proceeding in another way, isn't it? So, this method of expiring carbon dioxide, this product H2CO3 doesn't accumulate much, right? And the reaction can proceed towards the formation of the H2CO3. Okay. Then we have other buffer, that is phosphoric acid, which dissociates into hydrogen ions and phosphate ions. So, the pKa of this buffer is 6.86. Again, you see the pKa is much less than that of the plasma pH. So, more concentration of the dissociated ions will be there at physiological pH, like we saw in case of H2CO3 also. So, obviously this buffer will also be more effective for acids. Lastly, let us see how proteins act as buffers. Proteins have a carboxyl group and an amino group. So, carboxyl group can accept an hydrogen ion. See here, one hydrogen ion is added, thus preventing the change in pH due to the addition of strong acid, while the amino group can release hydrogen ion, and that's how it will prevent the change in pH caused due to the base, which is causing hydrogen depletion. So, one hydrogen ion can release from the amino group. So, what are the physiological buffers in body fluids? There is a phosphate buffer system which is important for intracellular fluid, and tubular fluid in kidneys. Then there is a bicarbonate buffer system, which is very important for extracellular fluid. And then there are proteins which are present both in intracellular fluid and extracellular fluid. In extracellular fluid, especially protein in hemoglobin, because hemoglobin has histidine amino acid, and since the concentration of hemoglobin is quite high in extracellular fluid, it forms a very important buffer in extracellular fluid. Then there is ammonia buffer system, again very important for tubules in kidneys. So that was about the physiological buffers and how these buffers act. Thank you. Have a nice day.